Hey guys, today we are going to take a look at variations on Mendel's idea of inheritance. So everything that we've talked about so far, whether we're looking at a monohybrid cross or a trihybrid cross, everything has been very straightforward dominant recessive relationships when we look at the alleles. So we're going to look at some variations on that. So we're going to focus on 5 different things: incomplete dominance, codominance, multiple alleles, epistasis, and then finally polygenic inheritance. So we'll start with incomplete dominance, and this is when the F1 hybrids show a phenotype that is somewhere between the phenotypes of the two parents. So we often call this a dose effect, and a really good example is with snapdragons. So these are flowers. You see actually 3 different colors of these flowers, but if you were to cross a red with a white, you will actually get an F1 generation that is all pink. Then if you cross the pink, you'll get an F2 generation that shows a 1 to 2 to 1 ratio of 1 red to 2 pink to 1 white. So if we take a look at this, here's our P generation. So again, we have our red and our white. I do want you guys to notice that the alleles are written separately. So here we have C capital R's and C capital W's. Written as with the R's and W's written as a superscript. And so C here would be color, and the red is the R, and then the white is the W. And so when you cross the red and the white, you get the gametes CR and CW. So you will get an F1 generation of all pink flowers. And then when you cross 2 F1 individuals with each other, you will then get the Punnett square shown below with the following ratios. So you will have 1/4 of them you would expect to be red with CR, CR. 1/4 you would expect to be white, CW, CW. And then you would expect half to be pink again with the heterozygote genotype. So that is your F2 generation with incomplete dominance. So the result of this is that you have no hidden recessive. You have 3 possible phenotypes and 3 possible genotypes. And so again, we tend to see a dose effect. So if you are heterozygous, you show the intermediate of the 2 colors, and then you can be homozygous red, homozygous white, or heterozygous, which will be pink. All right, now codominance. This is when both alleles are expressed equally in the phenotype. The way that I've always thought about codominance is think about a coworker. When I come to work with my coworkers, we are both here. We don't become blended into the same person. So with coworkers, both individuals show up with codominance. Both alleles will show up in the phenotype. So both alleles are expressed equally in the phenotype. An example is the MN blood group. So you can have homozygous for M, homozygous for N, or you can have the heterozygous, which is M and N, in which case both will show up. So again, you have no hidden recessive. You have 3 possible genotypes along with the 3 possible phenotypes, but it is not a dose effect. It's not you're not blending the M and the N together. You simply have both showing up. Another example is with chickens. You can have black feathers, you can have white feathers. Those are the 2 homozygous forms, so homozygous black, homozygous white. When you have the heterozygous form, you don't get gray feathers. What you get is a mix of black and white feathers on the same bird. All right, so it is not a dose effect. It's just that both, essentially both phenotypes are being shown. All right, next we have multiple alleles. And so this is when there are more than 2 alleles for a particular trait. So a really good example is the ABO blood group. And so we have 3 possible alleles for the ABO blood group. The first is written capital I with an A superscript. This codes for the A antigen. Then you have capital I with the B superscript. That's your B antigen. And then you have a lowercase I, and this means that there is no antigen. So these are your 3 possible alleles. Again, even though there are 3 possible alleles, every individual will only have 2 of these 3 alleles. So the result of this is that you have multiple genotypes and phenotypes possible. And this is a very common event with many traits. But when you look specifically at blood types, if you are considered type A blood, there are actually 2 genotypes that could code for type A blood. So you could be homozygous for the IA, or you can be heterozygous and have an IA and a little I, in which case you're going to have type A blood. You could be type B blood and it would be the same thing. You would have homozygous for the IB, or you would have the IB and the recessive I. Both are type B. You could also have type AB blood, in which case you are heterozygous, and so you have one IA and one IB. Or you can have type O blood, in which case you have two of the recessive little I alleles. So you can see here that with the multiple alleles, you have way more options for genotypes and for phenotypes. This is just a diagram sort of breaking that down. It goes into a little bit more detail about what we mean by these A's and B's, and these are different antigens that appear on the outside of red blood cells. And so when you have the A antigen and you are part of the A blood group, that's your phenotype, you have two possible genotypes. When you have the B antigens and you are the B blood group, again, you have two possible genotypes. If you are the AB blood group, then there is only one possible genotype. If you are the O phenotype, again, there's only one possible genotype. So just keep in mind that the Rh blood factor is a separate factor from the ABO blood group, so that's where you have the Rh plus, which is dominant, Rh negative, which is recessive. And so when you're talking about like A plus blood or A positive blood, that is actually a dihybrid trait, and we were only focusing on that example on just if you were type A, type B, type AB, or type O, regardless of the positive or negative. All right, next we have epistasis, and this is when one gene locus alters the expression of a second locus. So one gene will alter how a second gene is expressed. So we're going to be looking at an example of fur color. And now the first gene is C, and so the C is for color. And so the dominant form means that the fur will have a color. The recessive form is albino, meaning that there's not going to be color to the fur. Then the second gene codes for what color will be there if there is color. And so capital B is for brown, that is dominant, and then lowercase b is for black, which is recessive. And so this example, we're looking at gerbils. You can see here we've got several different colors. So this would be our albino gerbil. We've got a black gerbil, and then some different shades of brown in there as well. So if we look at the genotype that is heterozygous for both color, the presence of color, but also then the actual fur color. We look at these two individuals. They do have the same genotype. They are both brown. The reason they are both brown is because they both have at least one capital C, which means that their fur will have color, and they both have at least one capital B, which codes for brown fur color. Now, when these two are crossed, what you end up with is a 9:3:4 ratio, which is a little bit different than the expected 9:3:3:1 ratio that you get in a typical Mendelian dihybrid cross. So we have 9 that are brown because there are going to be 9 individuals that have the capital C and the capital B, meaning color in the fur, and that color is brown. We will have 3 that are black because they have a capital C, meaning that the fur will have color, but they are homozygous recessive, so they will be black. And then we have 4 that are going to be albino. And they will be albino because they inherited 2 recessive Cs, and so there is not going to be any fur color. They will be white. And so you can see here that this the last 2 letters have been left blank because honestly it doesn't matter. Their fur is not going to show up black or brown, so you can simply write it as lower C because they will be albino. And so this part of the genotype right here essentially blocks this part of the genotype from being shown in the gerbils. So you can't actually tell, if you were just looking at an albino gerbil, you wouldn't be able to tell what these alleles are because you can't see that within the phenotype. So the result of epistasis is that ratios are often altered from what you would expect. One trait may act as a recessive because it's hidden by the second trait. And so again, that albinism alleles, when you had the 2 recessives, it hid the fur color alleles. You couldn't tell what the mice were, or what the gerbils or mice were Again, we have the same example in mice. So we've got the alleles for color, and then also the alleles for whether or not the color will show up or if it will be albino. So again, we can see here 9 to 3 to 4, although in mice, capital B is for black fur, and then the lowercase b is for brown fur, so that part is different between mice and gerbils. So let's do a quick practice problem. Let's say you have a wife who is type A blood, and you have a husband who is type AB blood. They have a kid. This child has type O blood. Is this possible? So if the wife is type A, there are 2 possible genotypes that she could have. If the husband is type AB, there's only 1 possible genotype. So if I told you that the wife's boss is type O, does that change your opinion of this situation or the wife in question? Before you guys jump to any conclusions, though, I want to talk about something called the Bombay effect. And this is an epistatic gene on the ABO group. And what it does is it alters the expected ABO outcome. And so here you have a capital H, which is dominant, which will allow for the normal ABO blood group. Or you can have the recessive H, which means that the A or B will not show up, and it reads as type O blood. So this gene with the H's, those alleles, will affect how the ABO alleles show up. So if you look at this and you look at both genes together, and you see that the wife is type A and she's actually homozygous for type A, and then she's heterozygous for the H's, and if you look at the husband who is type AB, so he's obviously heterozygous, and he is also heterozygous for the H's, what that means is that the child could be IAIA or the child could be IAIB, but if he got 2 little H's, it means that he's still going to show up no matter what with type O blood. So therefore the child is the offspring of the wife and her husband and not the boss. Okay, so this is the Bombay effect, and this is going to be detected when the ABO blood type is different from what you would expect, like in that last case. And now we're going to talk really quickly about polygenic inheritance. Poly meaning many, genic referring to genes. And so these are often factors that are expressed as a continuous variation. So there are not clear boundaries between the classes of phenotypes. So 2 really common examples are going to be skin color in humans as well as height. So you are not like black or white and there are only 2 skin colors. There is a range of skin colors. Along with height, you are not, 5 foot tall or 6'5" and nowhere in between. You have a very continuous scale of height as well as skin color, and so there are not very clear boundaries between these phenotypic classes. And so when you have polygenic traits, you generally have several genes that are governing the inheritance of that trait. So again, skin color is likely controlled by at least 4 genes, and each time you have a dominant allele for one of those 4 genes, it will lead to slightly darker skin. So if we look at this example here, we have a bell curve of skin color. So if you look at the really pale side, essentially you have no dominance. Okay, so these would all be recessives leading to really pale skin. If you look at the darker end of the bell curve, you have more dominant alleles, and so what that is it's leading to a darker skin color. Then right in the middle, it's half and half, and so again, it causes a continuum of phenotypes, not discrete categories. So again, the result is that Mendelian ratios fail, traits tend to run in families, and offspring can oftentimes be more of an intermediate between the parental types. And just like we looked at, a lot of times it will show a bell curve and continuous variation. All right, so that's it for today. Your lecture question is, what other human traits would you think are polygenic and why? So think about human traits that tend to have a continuum, and shoot me an answer via the class website with any trait in a human that you think could be polygenic and why you believe that to be so. All right, thanks guys.